and the second one will be about the integrated proteomics pipeline. Today's presentations are scheduled to end at 9, so each presentation will be about 30 minutes each. Again, these conferences are recorded and can be accessible online once we've processed them. If you have any questions during the presentation, please type it in the message box provided in the application. And also remember to mute your mic during the presentation to prevent any unnecessary sounds. Are there any questions before we begin? Okay. So our first presentation will be presented by Yasset Perez-Riverol. Yasset was graduated in 2006 from software engineering at Polytechnic, Havana, Cuba. He worked as an associate researcher at the Center of Genetic Engineering and Biotechnology in Cuba from 2006 to 2013. He finished his PhD in biochemistry in 2013 at the Havana University. In March 2014, he moved to the PRIDE team to work as a bioinformatician with focus in mass spectrometry resources and tools. And today he'll be presenting about the expression interface, which will support researchers by making a distributed search of protein expression data through heterogeneous resources like PRIDE, Peptide Atlas, and PASSIVE. Without further ado, allow me to introduce Yasset.
Thank you, Vincent. Do you hear me?
Yeah.
Yes.
Okay. Well, thank you to all of you to be here. I will try to present in a short way the idea of PROteomeXchange expression interface. Okay. The current situation of expression data in repositories is an increasing amount of expression information is already deposited in different repositories such as PRIDE, GPM-DB, Massive, Peptide Atlas, COPAC-AB, and PROteomeXDB, for example. And this number of resources are growing so fast in the recent year, in the last couple of 2 or 3 years. Now the necessity of web service has become generally accepted. They are widely used in repositories. This is the current situation. And biologists and biological resources are interested in.
Yes. Excuse me. I don't think your slides are displaying.
Sorry?
Are you changing slides?
Yeah.
Oh, they don't. You're still on the first slide.
Oh, it's not.
It's not in presentation mode.
Do you see here?
We still see the first slide.
Okay, sorry. Let's start again. Can you share again?
Okay.
Sorry. Okay. Okay. Now?
Yes. Now we see there. There we are on the first slide, full screen.
Yeah.
Yeah.
Looks good.
Thank you.
All right. Thanks.
Okay. Thank you. Sorry about that. Okay. Now it's a common practice that biologists, as I said, biological resources are interested in proteomic expression evidence at different levels, from protein to also view on a specific data set that, in our experience in PRIDE, is a common practice that user access to different data sets, individual data sets, to retrieve the protein information, also the raw file, the spectral file. Why we need something like a protein expression interface? Here are the more common resources nowadays, and if you see here, most of them appear in the last 2 years or something. And some of them, for example, are PRIDE, as I said, Massive, Peptide Atlas that belongs to PROteomeXchange consortium, but others like MOPET, ProteomicsDB, MassQB, and COPAKB are also really important in this context. Changing proteomic results need to make proteomic data accessible and very easily accessible by different resources. We have the necessity to share proteomic results with researchers outside proteomics, which means that make easy to access to this data without big knowledge about the data formats and the insights inside proteomics, like search engines or scores. And proteomic resources also require mechanisms to simplify and efficiently exchange basic proteomic results, such as identify, protein, and quantify expression data. But more important than that, we need to integrate different distributed resources and different pipelines from well-established repositories to also group data sets. Bioinformatics groups that they don't have a really big support in terms of developers, and to be able to create their own repositories and share their data. With protein expression interface, we will try to learn from previous experience, especially from Scikit. The approach, Scikit was published in 2012 in Nature Method, and is a repository. It's the same system for interactions data, and the idea is that you have different interaction databases, and on top of that you have Scikit implementations and an API, and then you have different clients to access to these databases. You have a common registry where you will see all the databases that implement Scikit, and then you can do normal queries in a web page and retrieve the information from different resources. On top of that, you can implement different web client can be web client or command line clients and different tools for analysis. What is the PROXI aim? Is to provide a simple and efficient way to exchange protein expression data using distributed repository, protein and peptide that was identified in a given experiment setting, which protein and peptide identify in different experiments. We are planning to implement something. It must be really simple and easy to implement and they maintain. And we are trying to base everything in a way that it can be robust and also to make accessible data from these different resources using simple tab delimiter file output or JSON objects. As I explained before, the idea is to have different databases like PRI, peptide, like HPA or GPMDB, with a PROXI implementation on top of that, and then we can build different clients and a PROXI registry. The idea with the registry is to have all of the implementation of PROXI in a central point that you can basically query this central point and see which of the services are active or not active and which kind of information they contain. The concept also right now propose different view of the data. We are starting from dataset view, and it's really important, for example, for resources like PRIDE to have this kind of view. It must be report result related with one experimental unit, for example, PRIDE projects, where you have different data sets, and then you can see in this data set especially which protein was identified and which peptides. Also, we are planning to implement the protein view that can be an aggregation. It can be seen as an aggregation of different experimental units, keeping in mind protein as a central point of aggregation. To understand this concept is like peptide atlas. When you go to peptide atlas, for example, and you try to retrieve information on one particular protein, you can see all the experiments where this protein has been seen, and also all the peptides that were identified for this protein in different experiments and so on. And also the last one will be have something like an expression view, that will enable to retrieve information of quantitation value of different protein and peptide across tissues, etc. This is etc. should contain minimal information for proteomics repository knowledge and basic to exchange data like protein information, peptides, and metadata. What the service does not aim at this point is to replace the repository, proteomic resource, and API. It means that every repository can also have their own API and web service to provide their data, but with this API, I mean the API belongs to these repositories. They can provide more data in a different way that we are providing in PROXI, but at the same time they can run the PROXI view for their data. It will mean that we will in this central point we will try to generate also more traffic to those resources. And we will not keep any. We will not try to retrieve data and put it in another place like a central point. That the idea is to have a distributed system. This is. I will try to explain some concept, but here you will see the PSI Proxy GitHub web page where we are just starting to documenting the API and the schema, but also we have already a web page to explain the whole concept and also to highlight which are the partners involved. For every resource, you will have something like a URL, and then after that, you will have the version of the PROXI implementation. In our experience with the Scikit, you can have different versions of the same PROXI specification with different methods. And if one resource wants to keep this version in mind, then they can have a different version of the API running at the same time. Then you will have the resource data, for example, proteins, and then one ID for each protein to retrieve the information of the proteins. As I said, we have already a web page when we have in GitHub when we have all the schema and the use case that we are starting to implement. For example, the protein schema contains one accession that is the resource accession. We try to keep as small as possible the accession that every resource is using independently. If they are using, they can use UniProt or Ensemble. For example, Petra Atlas normally use UniProt when GPMDB like to use an Ensemble ID. But at the same time, we are trying to encourage that it can that they added the UniProt and Ensemble IDs to each proteins. Then we are trying to, for example, to put synonyms to each protein that can be used. For example, if you have other IDs for cross-reference to different resources, the sequence of the protein and the reliability of the protein, which is related with how many times this protein has been observed in the repository and can be used as a different metrics, and the number of observations. This object is huge. I mean, contain other other properties, and you can only, if you are interested, you can go to the GitHub, as I said, to the GitHub webpage and see the schema of this protein entity. Also, we already defined the schema for the peptides, for the modification, and so on. Well, the progress of the project and the website, you can go to the website, which is PSI Proxy GitHub. We are trying to run everything in GitHub from the implementation detail, but also we are trying to put all the documentation in GitHub and trying to follow the project in GitHub, like creating the use case, closing issues, opening issues. Also, we have the repository itself when you can find the schema, the API, and the reference implementation. We are planning to implement the reference implementation for those groups that do not have a bioinformatics support, and they have their files, for example, in MC.ML. Perhaps we will explain this in the future, and they will be able to index their files and have their own and run their own proxy servers. If you are interested to go to the API and the schema, this is the URL for the API and the schema, and see the issues of the priorities that we already started to work on. Okay. Acknowledgement, well, this is a project that is running in Proteom, belongs to the PROteomeXchange umbrella, and the developers and the teams that have been involved on that are the PRI team, led by Henning and Wanan, and the Peptide Atlas and the massive group, like Eric and Nuno. Questions? Thank you.
Thank you, Yasset. That was very interesting. If there are any questions from the audience, you can just speak out or type them into the chat box.
I have a question. So for the GitHub, is it like a public access, or do we have to get permission to access your API or code?
Well, this is a really nice question. Actually, right now, to contribute with the code, we need to get everything is open. You can see the progress of the project, but if you are interested to contribute, which is actually really, we are interested on that, you can basically send me your user, and I can invite you and add and you can add issues or even contribute coding and the schema and discuss the schema of the objects and the API itself.
So this is Andrew, so I have another question. So this may be obvious to people in the proteomics community, but can you walk through who you envision the PROXI clients being and what the use cases are there?
Well, for them, the basic for the proteomics community, for example, the basic use case is to see all the protein that has been seen in different resources. Nowadays, we published last year a review about how to integrate all of this data, how to integrate all of these protein identifier, all of these protein coverage. It's practically impossible to merge all of the resources that are around. I mean, like Peptide Atlas, EMBL-EBI. If you try to merge all of this data right now, it's practically impossible. For example, for resources like NextProt or UniProt, which are knowledge-based database, they need to consume this information, like in which particular experiment this protein has been seen, which are the peptide that has been identified, how many times this peptide has been identified, which modifications. Nowadays, they need to download the files, reprocess in their own pipeline, and sometimes convert between different files. It's a really like a nightmare, I think. And in top of that, you can build a lot of things. For example, the first thing is to show different expression value. How many, quantitation value between different experiments, between different tissues, disease, okay? And the other thing is, which is more and more interesting, is to validate results across different resources. I mean, if one particular peptide has been seen in one particular experiment, and this particular peptide has been seen also in a different experiment, and this can be used to to like a high quality or measure of the identification. We are including, if I have some time, we already include some scores like reliability score that it follows this approach. I mean, trying to see how many times one particular peptide or one particular protein has been seen in different experiments, and try to validate across different experiments. As you know, for example, Peptide Atlas use their own search engine, their own pipeline. GPMDB use a different pipeline, a different search engine, and this can be really like a big improvement in the way of doing this. Also, for example, well, all of the things related with visualization of the data and modifications. There is a lot of use case we can implement. Right now, we are only trying to define and standardize the way that we are providing this data. Okay.
Great, makes perfect sense. Thanks for that explanation. Just two quick thoughts on that. So first, I mean, I think it is, as you define the specification, I think it probably is good to have one client partner that, will be the consumer, at least initially. Otherwise, you finish the specification and you don't really know the uptake and things like that. The other thing that comes to mind is that it a little bit has the same goals of the Global Alliance for genomic health, right? What they're trying to do for genomic data sounds very similar to what you're trying to do for proteomic data. I wouldn't suggest, merging them or trying to be too generic in that regard. But if you aren't already sort of keeping an eye on what they're doing and how they're defining their schema in terms of data exchange for genomic data, I think is probably worthwhile.
Well, yeah, this is a really, really interesting. I mean, we are not in the moment that where they are in terms of they are trying to release something for big data sharing, but more for not for the evidence they found, also for the raw data. You mean in terms of proteomics? But we are more interested in something on top of that, like the repository we already have and the way to exchange this data. Perhaps in the future it will be interesting to go further on this and then trying to share, for example, Spectral data or PSN data to do cross-validation between different experiments and so on. But yeah, I know about this experiment, about this project, and actually I'm following them. But yeah, it's thank you for the advice. I mean, yeah.
If I may chip in on, this is Henning, on two of the use cases. So one thing we already have in a raw form is to just show expression data in the reactome pathway analysis. And in this case, reactome is basically a client, or would be, and in the future would be a client to PROXI. So, and in a similar way to what is already now implemented, where on reactome we can show interaction data, and so reactome is a client to Psychic. So this is one thing. And the other on a per protein basis. As an example, at the moment, the UPO HPP project is very laboriously generating the list of all the proteins which have been seen, or the human proteins which have been seen, and what the evidence is. And a vision, which I would hope to achieve at some point, is that this is not a lengthy operation where you have to integrate many files and update manually whenever one of the sources is updated, but this is basically something where with real up-to-date data from all relevant sources, you would see this at any given time point via PROXI.
Great, that sounds, yeah, perfect. Very useful contact. Sorry, one thing that occurs to me also too, while we're, we touched on Global Alliance, right? I mean, I think it's interesting. I would be interested to know in the data repositories, right, how much clinical data you have. Is there the equivalent of, dbGaP or clinically relevant proteomics data? Because that, then you also could leverage Global Alliance's like infrastructure on, privacy controls and things like that. But I mean, I think that would just be, a useful thing looking forward as well. How do we share clinically relevant proteomics data between different groups?
I agree. I think this is something which will come up over the course of this project. To my knowledge, so far there's not the equivalent of, let's say, explicitly shared but with controlled access large-scale proteomics data, but I think this will come about, and I'm not sure we'll manage to integrate controlled access strategies into proxy in this round, but at least, yes, this would be a good vision to be able to do this, to integrate for those who have access more data than for those who don't.
If I have Vincent, I still have some minutes, or?
Yeah, hi. What were you asking?
Well, yeah, because I can show you, for example, the web page, and I don't know if you've seen the web page.
Yeah, you can show it to us.
Okay, this is the web page. Is running in GitHub, and that's why I said it's a GitHub-oriented project. And then you will see very quick the schema, all the schema that we are now generating, the API information, and our GitHub repository, and the partners that are involved. All of them, well, the first one like Brian, Peptide Atlas, and Massive, and Puzzle are PROteomeXchange, and COPAC-AB. I have been talking with Vincent about the integration of the API from COPAC-AB with PROXI. And also, we have some information about the use case that you can basically start we are planning to start from the very beginning. And if you go to the API, let's see if it's working, the API, the general concept of the API, the URL syntax, and what is our view of a protein, the relation with the modification peptides variant, and so on. The vision for the data set, and we are starting to put more and more information here. Also, we have some documentation, like for example, what is the main idea of about proxy, what are the claims, and what is the aim of the project, and so on. If you are interested to contribute, basically send me your user account, GitHub user account, and I will give you access to it. Okay.
Okay, sounds good. Anyone have any more questions before we move on to our second presentation? Okay, thank you, Yasset, for that great presentation. We'll move on to our second presentation. So our second presentation will be presented by Robin Park. Robin is a bioinformatics staff at the Yates Lab at Scripps and the co-founder of Integrated Proteomics Applications. He was previously employed by ActiveX Biosciences in San Diego, and then he worked at the Biomedical Informatics Research Network at the University of California, San Diego. During his 10 years at Scripps, Robin has published a number of scientific publications, books, and has given many presentations at the proteomics conferences. Today he will be talking about the Integrated Proteomics Pipeline, which is a proteomics data analysis platform. Without further ado, allow me to introduce Robin.
Hi, this is Robin. Can you see my screen?
Hold on.
Okay. Give me a second. Or show my screen. Okay. How about now?
No,
Not yet.
Yeah, I see your screen.
Oh.
Yeah, so.
Okay. Can you see now?
Yeah, looks good.
Okay, good. This is Robin. So here the IP2 is the comprehensive proteomics data analysis platform. So we are really working hard to expand the IP2 to expand its features and also keep up with the latest proteomics technology. And today we have a pretty limited time, so I cannot cover all features or background behind the algorithm, but I can just pinpoint some key features of IP2. Meanwhile, next month we are going to have an IP2 workshop at Burnham, San Diego. So we are going to discuss more in detail about the IP2 there. So this is an overview of the proteomics data analysis workflow. So we have a raw file, and then we can extract the raw file by deconvoluting or DI stopping, and then we can generate MS1 or MS2 scans, and we search against the protein database. And then after identifying proteins, the next step is in general the quant analysis. There are a whole bunch of different quant analysis strategy, MS1 based like M15 or Sileg or dimethyl, the isobaric like TMT or iTrack, istopolog, and also the label-free analysis like spec count based or intensity based. And then the next step is the statistical comparison of multiple samples to find interesting proteins confidently, or you can also do the functional analysis, like it can be gene ontology or pathway analysis or protein network interaction. So this is an overview of IP2 pipeline. So IP2 is a web-based application. So user can access through the network by using web browser. They don't have to install any software. And IP2 behind it, we have many different softwares installed. And also backend, IP2 support many different type of backend layer. It can be a cloud computing, or if you already have some cluster, like a local cluster, then IP2 also can support many different type of cluster like SG, PBS, or some other, the Slurm or other types. If you don't have any of them behind, and if you your lab is just small, then you can have only single IP2 box, and then you can analyze everything inside IP2, in which can be slower, but it can be good enough for a small lab. So basically, in order to run IP2, all you need is to have one single computer. So we have some selected customers here, the scripts or Berkeley, Dow Chemical, and NIH, and we keep expanding more and more. So the goal of IP2 is that to build a one-stop data analysis platform. As the data is getting bigger and bigger, and also the way to analyze proteomics data is getting more and more complicated, we try to simplify the data analysis by clicking some button and quickly move to next step. And automation, and also we provide many different, graphical tools, so user can intuitively understand the result, and they can interpret the result quickly. And also we store data in one place, so that you don't have to worry about the losing data, keeping on your desktop and then crash your computer. Everything is stored in one place, and you can back up only that one file server. You can access data from anywhere, even you can use the iPad or the iPhone, and then when you travel, you can still continue to working on your project. We also provide many statistical tools. And also, if you have your own tool, or if you want to modify IP2, we provide IP2 development kit, which is called IDK. So we provide the PDF manual, so usually developer can follow step by step to figure out how to integrate any of third-party tool or change IP2. We also provide the tool to manage your project or data. When for example, the scripts, the EH lab, we have a huge amount of data for lasting some decades, and so we have many postdoc came and then gone, and all data are organized in IP2. And also you can share project with a collaborator. You can specify which project to share. So with IP2 you can again organize data and share project, and also you can do protein identification, and also pretty much IP2 can cover most quantitative analysis, and also again statistical tool and Pgm analysis and functional analysis tools. And also recently we started working on some new project. So on the cloud, even though IP2 can analyze all your data by using cloud resource, we are also going to make IP2 to transfer all your data to store in the cloud storage, like the Glacier or the S3. So the advantage of the cloud storage is that there's pretty much the largest storage space, and also you don't have to pretty much minimum maintenance is enough to keep your data. And also you can make your data public or private. So one of the features in IP2 is to have spectral quality control. So currently you can control the spectral quality, but we are going to expand that to the ID or quantity level. So there are many different features, and by clicking some button in IP2 you can see this is a part of the graph, but there are actually more. So you can see overview of the quality of your spectra. So you can when you have some problem, or you can make sure that your data from instrument are in good shape. So when you load the data, the first step is to identify the protein or peptide. So when you have a theoretical spectra and also experimental spectra, you basically compare ions in MS2 to see which one, how well they match, and then it calculates score. We compare this is a raw curve. We compare the different search engine, and PROTomics is the one of search engine in which is developed at John Yates Lab. And we just check how many true spectrum hit, and then we compare the different search engine. PROTomics generate the most ID, and then it followed by Sequest and other like Mascot and OMSA. Which are not included here, but we also compared with Comet and some other search engine. Still, PROTomics generated the most IDs. So currently, the PROTomics and they are the available search engine. So PROTomics again can generate most ID, but it's performance-wise, the Comet is somewhat faster than PROTomics, but you have to sacrifice a certain number of protein ID. In general, 10 or 20%, if it's big, well, it can be even bigger. And also, we have a broad-mass PTM, so basically it's a blind PTM search. Usually don't need to specify any PTM, and this software will automatically check, to find all possible PTM sites. This was developed at Yates Lab and by Lin. And another one is like a broad-mass, another search engine. When you have a very big database like RNA translated DB or meta-proteomics, the database can be pretty big, and then most search engines has some kind of difficulty to search. So this is like we have some unique algorithm, and this is developed by Yates Lab and Orland collaborator. And we tested a 70 gig database, and we successfully searched it. It took about 1 or 2 days in a single 16 core computer. Next one is isobaric labeling analysis. This is a typical isobaric labeling analysis overview. So when you have multiple samples, and then each one we have some TMT or iTrack tag, and then we combine. And then when you generate the MS scan, we don't see any difference. But when we fragment into MS MS, you can see all the report line from different samples, and by measuring the intensity of each one, you can see which samples are expressed. So this is a data analysis of TMT. When you have let's say the 6 different samples, and usually they put biological replicate, and then usually can specify how they are going to group the samples based on biological replicate, and then we normalize it, and also we IP2 can retrieve. IP2 is based on by using the sensors, it can retrieve all good quality scans. And also we can apply intensity filter. This is described in the Sense2 paper last year in a bioinformatics journal. And then it can compare peptide ratio between samples and also outlier filtering. And based on peptide ratio, we can calculate the protein ratio. And then next, the final step is to apply some statistical analysis. And also the fusion is pretty powerful for TMT analysis by using a multi-notch. So we also made IP2 to support multi-notch analysis based on MS3 scan. And also when you have HCD CID combined scan, then usually the CID can be fast for the scan. So CID can be used for ID, and HCD can be used for the quant, and then IP2 also support. That's a part of some the parameter. This is the final output of TMT analysis. So when you have this is usually actually in this case a group, the 3 report ion in one group, and then second another 3 in a second group, and then there are many different the values like standard deviation, relative standard deviation, and then it generates the p-test, p-value, or the q-value and q-value. The level-free analysis, there are 2 different level-free analysis. Again, one is just spectral count based, and second one is intensity based. So bottom of proteomics is the most the popular approach, but there is one drawback that you usually have to keep in mind. When they use a spectral count, there is always some isoform or subset protein issue, in which means sometimes you can see some missing spec count when you compare samples. SCP, that's one of the tools that developed at EHLab. It can retrieve all missing spectral count from unfiltered data. So this is one of the example. So in the regular search, this is some isoform or subset issue. So the one protein can be have a 0 value because this is the subset of some other isoform protein. And then we applied some new tool to generate to retrieve the missing spec count. And then this is a biological replicate, biological or technical replicate, from one of our projects. And ideally, they must be the same number. Theoretically, when you retrieve it, they are pretty much similar numbers. So we retrieved a lot of the missing spec count, in which help to calculate some statistical the p-value later on to find the regulated proteins. So this I think the blue line is the original spec count, and SCP retrieve the spec count. So each protein we can see the retrieved value is pretty much the same, similar to the original the replicate of a protein. So in which means they are working pretty well. And this is the volcano plot, and the size of the circle show how abundant that protein is. And label-free analysis using chromatogram. So when you have when you compare samples, it's usually you generate the replicate, either biological or technical replicate, and then when you do ID, and then align, and then extract the chromatogram, and then compare different samples. So our level-free analysis, we recommend to have high-resolution data to get confident the result. And also we do chromatogram alignment based on retention time. And also we used to identify the peptide to exclude any false ones. And also we, even though you couldn't identify some peptide, we can use other experiment replicates to retrieve that those missing peptides. Also ion injection time correction. And also we do global normalization. And also we also correct the mass any mass drift in the sample. Another feature in the IP2 is metabolic labeling analysis. So this is a pretty typical the labeling analysis, MS1 based labeling analysis. So when you have a labeled and a non-labeled sample, when you mix it, and then we measure MS1 based chromatogram. So this is the retention time, and then heavy and light, and then we check the correlation of heavy and light, and then we can check the quality of the peptide, and then if when it's in correlation to poor, then we discard. If it's good, then we keep it. And then this is a correlation score from these two comparison, and then the slope can be a ratio of each peptide. And then the next step is you can compare again the multiple samples to find the confident the regulated protein. This is upregulated and then downregulated, and that typically the working plot. This is confident score, and this is for the change. Another unique feature in IP2 is in M15 enrichment calculation. This is a very useful tool when you want, or when your project is about protein turnover, or when you use different some special enzyme. Basically, each residue contain nitrogen, so that every single the residue can be labeled. So this M15 the labeling can be very useful for such case. We also have a Pgm analysis tool. We are providing the localization tool by using A-score, and also we have a debunker developed by John H. Lab. It also can provide some confidence score for PTM site. This is each protein. We can see the overview of PTM site, the overall the location, and then we can also see what is the spec count for modified and non-modified, and what is the residue, neighbor residue around that PTM site. This is gene ontology. So with the gene ontology, we provided 3 different categories: biological process, molecular function, and cellular component. And we use the hypergeometric test for analyzing the p-value. And currently we are using the gene ontologizer and also the PSA Quant developed by the EH Lab. The Matthew is going to give a talk about PSA Quant soon. That's a gene set enrichment based analysis. We are going to integrate this one too. So these two tools are pretty much complementary. So user can specify which what is the organism, and by the first option you can use a whole the geo database as a background, and then your identified or quantified one as a study group, or you can use all identified genes as a background, and then study group can be only regulated one, and then you can run, and then you can generate a graph like that. And by default, it picked the 20 most significant the gene geo terms, but you can always download the all geo term, in which can be often pretty big. User can customize it. And also we have a Proteomics wiki. So not only IP2, but we also have some other the tools, bioinformatics tools. User can get more information. Yeah, that's pretty much. Hello.
Thank you, Robin. Are there any questions from the audience?
Robin, this is Andrew here. So I'm curious,
Yeah.
Do you have an estimate as to what percentage of people who use the online IP2 suite, what percentage of those people end up depositing their data in a public repository like PRIDE?
Oh, well, currently we don't have the tool to directly deposit to PRIDE or the cloud yet. Currently, we are actually putting all data at EHLab, the website. So we are putting in all data there for now. And PRIDE, we tried some years ago, and then we had some kind of technical problem, but we plan to transfer data again, but we haven't tested yet again.
Okay, I mean, that was exactly what I was sort of driving at. I mean, if there was a large number of people who I mean, that could just be a nice convenience function. But of course, I understand that many people who do analyses probably aren't ready, or maybe even eventually don't want to deposit in a public repository for whatever reason. So anyway, I just wanted to sort of get an idea of how much of that data eventually ended up in PRIDE.
Yeah, that's a good question. Actually, I got many requests from the users outside. So scripts, again, we just can put on to one of the scripts, the web server, and then we can put it on the web. So many journals are required to put the data in the public repository. So that's where that's how we are doing. But meanwhile, the IP2 users from outside scripts, they asked me, they requested some way to easily put the data into a repository. So my plan is to add some kind of easy button in IP2, so user can simply click the button and then data can be transferred automatically either to cloud storage or the PRIDE or some other the public repository. So that will be the ideal situation. A lot of people out there, they have still not very familiar with like a computer. They're not computer savvy, or they are not very. They don't want to go through all the manual how to figure out to transfer the data. So yeah, I want to make it pretty easy. Just one simple click button and then transfer the data.
There is an example of this which has recently been published from Leonard Martin's group, Peptide Shaker, which does pretty much that, and it can also, that's the other point, it can read data from PRIDE. So basically people can easily access data, run it through that pipeline, and then provide the results of this analysis, for example, because they've been hunting for specific modifications or have done some other kind of discovery. So I think it might be a nice project within BD2K to work on getting something like this going with IP2 too. So we would be very interested in doing that.
Did you say a pep shaker?
Peptide Shaker from Leonard Martin's group. It was a Nature Biotech publication which is just out.
Okay.
I think it was Nature Methods or Nature Biotech or Nature Methods. I'm not quite sure. I can send you the reference.
Okay, yeah, I can Google the peptide shaker. Okay, thank you.
So nobody else has another question, I'll ask another one. So Greg Stupp in my group has been working with Sandeep in the Wolin lab on this large-scale metaproteomics library searching, and in a different context, right? One of the things we proposed in our BD2K proposal was this idea of personalized genomics, right? Where you would not just be searching against protein libraries from the reference genome, but essentially translated from all genetic variants that have been observed into their predicted peptide variants and protein variants. And then as we got deeper into this, we weren't exactly sure what the use case would be here. So maybe this is just a discussion point for everybody. I mean, is this, what would be the utility of essentially being able to match your proteomic sequences against a larger database of peptides that included known genomic variants?
Yeah, I think that's a very interesting topic. Yeah, I think this can be, I think, the open discussion in this group. To me, I think that's a pretty useful tool, like just genomic data just to translate into proteomics data, and then we can, if the data base can be pretty big, and then we can search, or RNA-seq we can also translate, and then we can do that. I'm not sure at this moment how complete the how well how complete for proteomics the protein database, the FASTA database, and also the not only human, but human is pretty much human or human is pretty well annotated compared to other organisms. But still we have many the project that is we have a genome sequence, but there's no like a protein database or fully annotated database. So this approach can be pretty interesting the topic. So definitely our tool the plasmass can be used to run to find the sequence. Another idea is that we can also try the de novo sequencing, and then we can also compare with compare against compare to genome or RNA translated the database to improve the confidence. That can be another approach.
De novo sequencing on the proteomics level?
De novo sequencing on proteomics level, and then it will generate some peptide sequence, and so the de novo sequencing, the issue to me is that that's a good idea, but still there are so many false positives, but still they can be used to we can do de novo sequencing, and also we can also search against genome some genomics the translate the FASTA file, and then we can search, and then when we have two different results, and then we can compare to improve the confidence. They can be I don't know. I just did not just thought about that.
Well, if anybody has other additional thoughts on how because I think we have the technical infrastructure here to do it, and it's just a question of what we actually do with it in terms of detection of protein variants, of the sort of the proteogenomics thing that we heard about at the PI meeting, or if somehow that would result in better quantification. I don't know. We're sort of batting this around internally in my group right now. So if anybody has any thoughts, please get in touch.
Andrew, sorry, this is Sarah Scruggs. I had some audio issues. Would you mind please repeating your question?
The question that I asked, this is Andrew. Is that right, Sarah?
Yes. Sorry, I had audio issues and it cut out for a while and I missed it.
No problem. It was just the idea.
I wanted to think about your question as you requested, but I don't know what it is.
Yeah. The question related to how we do with sort of personalized proteomics, I guess, right? Right now, I guess most mass spec to peptide searches are based on a peptide library of the reference proteome, And so now in my group we've been playing around with a vastly expanded peptide library that incorporates known genetic variants. But then, so technically I think we can do it based off of some of the work that Robin, has already talked about. But the question is, what difference will this make? Being able to match peptides against sort of a personalized proteomics peptide library, what's the application? Does that actually matter? That's what we're trying to address.
Okay. Okay, thanks.
Are there any other questions or comments regarding Robin's presentation? Okay, if not, thank you, Robin, for that great presentation. We'll conclude the conference here.
Comment?
Yeah.
I don't know if anybody else has tried this so far, but all of this meeting I've been following on an Android phone client just using the phone data connection, and it works pretty well. I don't know if everybody's aware that this works. And it was no problem. I guess you also could hear me at least. I got answers to my comments. So that works.
Okay.
All right, thanks.
Yeah. So there's Android and iOS support for GoToMeeting, so you can access it through these mobile apps too. Yeah. So next week we'll have presentations about Sidekick and Pint. So that's next week, and also again, be on the lookout for an email regarding using Trello and GitHub to track our progress, and that'll be out sometime next week. But yeah, thank you all for coming. Brian and I will be on the line until 10 to take any questions or open for discussion. Yeah, but thank you.
Thank you.